Proteins part first. In this e-content, we will study about various functions of proteins, some important discoveries related to proteins, general structure of amino acid, different types of amino acids, and some important properties of amino acids. Let us start proteins part first. What are proteins? Proteins are the macromolecules consisting of one or more long chain of amino acid residues. In simple word, we can say that the proteins are the polymers which are formed by joining amino acids, and thus amino acids are the monomer. Functions of proteins. Protein is necessary for all living organisms and they occupy central position in the architecture and functioning of living matter. All enzymes are proteins. These enzymes catalyze the biochemical reactions in our body. An example of enzymatic reaction is glycolysis, which is completed in 10 steps, and each step requires a particular enzyme. The absence of single enzyme stops the process and energy from glucose cannot be obtained. Thus, enzymes are very important for the proper functioning of chemical reactions in our body. Another important function of proteins that they act as a receptors on cell membrane. Hormones, neurotransmitter and other signaling molecule bind to these receptors and convey signal to cells. In this way, protein plays a very important role in cell signaling. Some hormones are also proteins. The best example is insulin. Another proteinaceous hormone is glucagon. Both these hormones are required for the regulation of blood glucose level. Protein act as a transport channel in cell membranes. For example, sodium channel allow the passage of sodium ion. Potassium channel are specific for potassium ions only, whereas calcium channels are specific for calcium ions only. Proteins maintain the shape and structure of the cell. Cytoskeleton plays a very important role in this regard. Cytoskeleton is made up of several interlinked protein filaments. The proteins in the cytoskeleton organized in the form of microtubules, microfilaments and intermediate filament. Proteins are also involved in cell division. During cell division, the chromosome of a cell are divided into two halves by pulling apart. This separation of chromosome is done by spindle fibers which are made up of proteins. Proteins are needed for oxygen transport. Two important proteins play important role in the transport of oxygen are hemoglobin and myoglobin. Proteins are also involved in muscle contraction. We perform our daily life tasks such as walking, running, sitting, standing, writing, and even speaking only because of muscle contraction, which is due to contractile proteins. And the most important contractile proteins are actin, myosin, troponin, and tropomyosin. Protein protects our body against disease, and this function is performed by antibodies, which are also known as immunoglobulins. These proteins are produced in response to various disease and foreign particles which enter in our body, and they fight against the disease. Proteins control the expression of genes. Gene expression is a process by which information in a particular gene is copied in the form of messenger RNA, and then this messenger RNA form protein by the process of translation. This process of gene expression is controlled by various transcription factors. The transcription factor allow the transcription of genes of only those proteins that are currently required in the body. The transcription factors are proteinaceous in nature, and thus protein regulate their own biosynthesis. Discovery of protein. Protein was first discovered by the Dutch chemist G.J. Mulder, who referred it as a complex organic nitrogenous substance found in the cells of living beings. According to Mulder, this is the most important known substance in living matter, and without it, life would be impossible in our planet. The word protein was suggested by Berzelius to Mulder. This protein word is derived from proteus, which means preeminent or first. Franz Hofmeister and Hermann Emil Fischer was credited to the discovery of understanding of proteins as a polypeptide. Emil Fischer proposed the theory of peptide bond, the chemical linking by which all amino acids are joined together. James B. Sumner, who isolated and purified an enzyme urease from jack bean and confirmed the proteinaceous nature of enzyme, received Nobel Prize in Chemistry in 1946 for crystallizing the enzyme urease. Linus Pauling proposed the alpha helix model of polypeptide chain. This is the secondary structure of a protein. While Landestrom and Lang plays an important role in understanding the basic structural level of organization of protein. The various structural level of proteins are primary, secondary, tertiary, and quaternary. Frederick Sanger first of all determined the amino acid sequence of insulin protein in 1949. It was the first protein to be sequenced. And this demonstrates that the protein consisted of linear polymer of amino acids rather than the branch chain or colloids or cyclols. For this discovery, Sanger won Nobel Prize in 1958. In 1958 also, the first protein structure to be solved were hemoglobin and myoglobin by Max Perutz and John Kendrew. Now we know that the protein consists of many amino acids, and these amino acids join together to form a long chain, just like the beads are arranged in a string. Amino acids are thus regarded as the building blocks of proteins. These are organic compounds made up of carbon, hydrogen, nitrogen, oxygen, and sulfur is present in some of the amino acids. A linear chain of amino acid residue is called polypeptide chain. Amino acid structure. Amino acid is a nitrogenous compound having both the carboxyl group and amino group. R stands for side chain, which is different in different amino acid. The side chain may be simple as hydrogen atom, or it may be a methyl group, or it may be a more complex structure. In the structure of amino acid, we see that there are two carbon atoms. The first carbon is the carbon of carboxyl group, and the second carbon to which amino group is attached is called alpha carbon. The alpha carbon of most amino acid is joined by covalent bond to four different groups. On one side it is attached to amino group, other side it is attached to carboxyl group, R group on third side, and on fourth side it is attached to the hydrogen atom. And thus the alpha carbon in all amino acid except glycine is asymmetric. And because of this asymmetry, the amino acid except the glycine exist in two optical active forms. This is the structure of glycine amino acid. In glycine, the carbon is attached to two hydrogen atoms, and this creates symmetry, and thus in glycine the alpha carbon is symmetric. Two amino acid, isoleucine and threonine, have two asymmetric carbon atoms. Thus they have four optical isomers instead of two. Because of the asymmetry, the amino acids except the glycine exist in two optical active form, L amino acid and D amino acid. Those amino acids having NH2 group to the left, they are known as L forms or L amino acid, and those amino acids having NH2 group to the right are designated as D form or D amino acid. The amino acid found in proteins belong to L series. Many of the naturally occurring L amino acids rotate the plane of polarized light to the left. They are known as levorotatory. While other amino acids rotate the plane of polarized light to the right, they are known as dextrorotatory. Although more than 100 amino acids are present in various plants and animals, but only 20 amino acids are found as a constituent of most proteins. These 20 amino acids that are encoded directly by the codons of universal genetic code are called as standard amino acids. Due to universal use of these 20 amino acids in the synthesis of various proteins, Francis and Crick called these 20 amino acids as magic 20. These 20 amino acids are alanine, arginine, aspartic acid, asparagine, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine and valine. Non-standard amino acid. Non-standard amino acid, these are those amino acids that occur in living organisms but they are not found in proteins. The best example of non-standard amino acids are gamma carboxyglutamic acid and 4-hydroxyproline and 5-hydroxylysine. Gamma carboxyglutamic acid is a calcium binding amino acid residue and it is found in the blood clotting protein prothrombin, whereas 4-hydroxyproline and 5-hydroxylysine are found in the collagen protein. Collagen is the most abundant protein by mass in vertebrates. Non-protein amino acids. Amino acids which are never found as a constituent of protein but they play an important metabolic roles are known as non-protein amino acids. For example, L-ornithine and L-citrulline occur in free state in the animal tissue and are metabolic intermediates in the urea cycle. Peptide bond. The amino acids are linked to each other by a special type of amide bond which is known as peptide bond. And this peptide bond consists of carbon, oxygen, nitrogen and hydrogen atom. This peptide bond is also known as CONH bond. There is no freedom of rotation about C-N bond of peptide bond. The carboxyl group of one amino acid and the amino group of another amino acid take part in the formation of peptide bond. The amino acids are linked to each other by this peptide bond and they form a long chain called polypeptide. A short chain of amino acid consisting of two amino acids is known as dipeptide, while a short chain consists of three amino acids is known as tripeptide. A short chain consists of up to 10 amino acids is known as oligopeptide. Each amino acid in a chain is called as residue. A polypeptide chain consists of more than 100 amino acids. The two ends of a peptide chain are named as N-terminus and C-terminus. The terminal amino acid with the free ammonia group is called N-terminus amino acid and the other side of polypeptide chain with the free carboxyl group at the other end is known as C-terminus amino acid. Amphoteric properties of amino acid. What are amphoteric substances? Amphoteric substances are those substances which act as acid or base. They are acid when they donate protons and they are base when they accept protons. Amino acid reacts with both the acid and base, hence they are amphoteric in nature. In strongly acidic condition, the amino acid behaves as a base. The COO- ions gain a proton and it becomes a positive ion. Whereas in strongly alkaline condition, the amino acid behaves as an acid. The ammonium ion of amino acid loses a proton and it becomes a negative ion. Jitter ion. Jitter is a German word which means hermaphrodite. It is a molecule that contains equal number of positively charged functional group and negatively charged functional group. Amino acid has the ability to exist as a Jeter ion. At certain pH, which is different for each amino acid, nearly all amino acids exist as a Jeter ion. The alpha carboxyl group and alpha amino group of amino acids are ionized at physiological pH and they form deprotonated COO minus ion which bears negative charge, and the protonated amino group NH3 plus which bears a positive charge. An amino acid in its dipolar state is called a Jeter ion. Isoelectric point. The isoelectric point is the pH at which a molecule carries no net electric charge. Although the charge are present, but they are balanced, thus we say that the net charge is 0. At isoelectric point, the amino acid does not migrate in an electric field. This means that at this pH, the amino acid exists as a Jeter ion form. Different amino acids have a different isoelectric point. Similarly, different proteins which have a different combination of amino acid, they have a different isoelectric point. And with the help of this, we can separate proteins from one another and thus purify the protein. The dissociable alpha carboxylic group of amino acid and alpha amino groups are responsible for characteristic 2 pK values of an amino acid. The pK value is the notation for equilibrium constant for ionization. pK1 for COH and pK2 for NH3 plus of an amino acid. This is the picture shows the titration curve of amino acid alanine. At a very acidic pH below pKa1, which is about 2.34 in case of alanine, all the molecules of or all the amino acid have an overall positive charge. While at a very basic pH above pKa2, which is about 9.69 for alanine, the amino acid will have an overall negative charge. At pH near about 6, the amino acid exists as a Jeter ion. Thus we see that the amino acid behaves as acidic, behaves as a basic at different pH, and at pH 6 they exist as a Jeter ion. The isoelectric points depend on the structure of the side chain of amino acid. Different amino acid have a different R group or side chain. Acidic amino acid have isoelectric pH near about 3, while basic amino acid have isoelectric pH near about 9. Neutral amino acids have isoelectric pH slightly acidic and it ranges from 5 to 6. Thank you.